of terms in biochemistry, and it can get overwhelming. And it's not like you know every term, but as long as you know the word roots and the prefixes and suffixes and things, you can often figure out what words mean, even if you've never seen them before. So today I want to go over some of the key word parts that come up over and over again in biochemistry, as well as some examples of where you see them. I will also share the link to this document and my blog, where you can find this kind of like cheat sheet of what these different roots and stuff mean.
This is a non-exhaustive list, and it's a working list, so I'm going to keep adding to it as these things pop into my head. But let's start off with the first things first. Let's talk about prefixes. So it's important to know like the metric prefixes, so things like micro, nano, pico, deci, centi, all of that various stuff. And I have more on that in another post. But let's go and talk about what else you need to know. So there's some other number type things. When we talk about mono, that's like single or one. So we often use this term monomer, and this is referring to, well, it could be referred to a couple different things. So it could be like the single unit of like some things that can link up into polymers. So for example, glucose is a monomer of more complex sugars, and you have nucleotides, which are the monomers of nucleic acids, and amino acids, which are the monomers of peptides. You can also talk about a monomer when it just have like a single copy of a protein working by itself and not needing any other helpers, or not needing it and not working with like another protein. If it was working with another protein, we could call it a dimer. So di and bi, these mean two, tri is three, tetra, quartr, four, pent, five, six is hex, seven is sept, eight is oct, nine is nine, and 10 is deci. And then once you get higher up, you might see some of the times things used above that, but typically when you get into those bigger numbers, you're starting to, but you still don't have that many, then we use this term oligo. So oligo means few or little. You might have heard the term, just like people use the term oligo when they mean that as a shorthand for oligonucleotide. So these are often like when you're ordering primers for PCR. So an oligonucleotide, that would be a short nucleic acid chain, or an oligopeptide would be a short chain of amino acids. Now, if you get a lot, now we're talking many. We're talking polys. Poly means many. And so a polymer is going to be a long chain of linked together monomers, such as a polypeptide, which is a long chain of amino acids, which we refer to more commonly as a protein, especially when it folds up into its nice, pretty shape that can do cool things. Speaking of things, pleio means more. And something that's pleiotropic is going to be doing or affecting multiple things. And so this is potentially a drug doing more than you want. And so think you go through this like smart drug design strategy so that you design something to do one thing, and then you find out that it's actually affecting a lot of different things. We would say those were pleiotropic. It had pleiotropic effects. So it's affecting more things than just one. Okay, now let's talk about kind of location. Hypo, below, remember that rhymes. So hypo is under and below. So hypoactive, this would be like less active than normal. So maybe your receptor is not responding as much as normally, so it's like hypoactive. Or hypotonic, having lower tonicity. So tonicity is why things are going to, like liquids move from one place to another because of how much the concentrations in various parts and things, and more than other posts. But hypotonic is a term that you would see used. And if something is hypotonic, it has a lower tonicity, and so liquid would move out of that into something that is hypertonic. And so remember hyper, over. So hyper means over or above, and so we can talk about something being hypertonic. Or we could talk about something being hyperactive, like more active than normal. Another over above term is epi. So you might see this with like epigenetics. This is the field of study that deals with genetic information above the level of just the DNA sequence, such as things like DNA modifications, histone methylation, and things like that. So epigenetics, above the normal, just above just the genome, above just the sequence. Let's go below. Infra. So infra, you may have heard of infrared. That's like wavelengths of light with frequencies below those of red light. Go the other way, we get to ultra, above. So like ultraviolet, that'd be wavelengths of light with frequencies above those of violet light. Or an ultracentrifuge, which is a centrifuge that spins at speeds above those of a conventional centrifuge. And these are really awesome, and they spin really, really fast. And so they're above normal, they're ultra. Another word meaning above, over, beyond. We have a lot of words that mean like similar things. And so if you see any of these, it's probably going to be meaning something similar, but we can put it onto different words. And sometimes they have slightly different meanings. So supra, this one's like beyond, over, or above. You might see the term like supramolecular, so made up of multiple molecules. So not just a single molecule, but multiple things held together by non-covalent bonds. Okay, meso, middle or moderate. So something that's mesothermic is going to basically be living at a moderate temperature, and we'll get into later how therm is referring to temperature. Iso, this is equal or similar. So things that are isotonic would have equal tonicity. So we talked before about hyper and being over and hypo being below, and iso is equal or similar. So similar. I so don't care because what's happening because they're similar both places. Isomers are going to be molecules that are similar, but they differ in some ways, such as having different shapes. Okay, ortho. This means normal, straight, or correct. So you might see the term orthosteric referring to when a molecule is binding in the same site as like normal binding partner binds. So you might see an orthosteric inhibitor is going to be a competitive inhibitor that binds to like the active site of an enzyme where the substrate normally binds. So that would be ortho. Then you have allo. This is other. So this is like the opposite of ortho. Here it's binding at another site, so not that normal one. Or it could refer to something happening at one site that has effects at another site. And so you commonly see this when we're talking about like proteins and like conformational changes, so like shape shifts, how you can have an allosteric effect where something happens at one site where maybe something binds, and then you get this kind of ripple effect where something else happens on the other at a different place. Okay, so let's talk more about where things are. Endo and ento, these are going to be inside. So remember inside, endo, ento. A common place is endogenous. So this is something that's made by an organism rather than taken in from outside, such as hormones that your body produces and uses. This is an opposite of exo and ecto. So remember exo, external. So these are basically coming from outside. So something that an exogenous compound would be something that's added artificially or from the outside, such as a pharmaceutical drug someone takes or a recreational drug. These would both be exogenous because it's not being made by the thing itself. Speaking of self, auto means self. And so an autotroph is an organism able to make its own food. And so we'll see later, troph refers to like nourishment and eating and things. Inter, this means between. So INTER, like intermolecular interactions are going to be interactions between different molecules. Don't confuse this with intra. So INTRA, this means within. So basically, intramolecular interactions are interactions between different parts of the same molecule, as opposed to inter, where it's happening between different molecules. You can also talk cis and trans. So trans means across. So something like a transmembrane protein is going to go across or span membranes. We talk about cis, this is going to be same. So like a cis regulatory elements would be a regulatory like non-coding region of DNA, such as a promoter that's on the same piece of DNA and like nearby the gene and it actually regulates the expression of that. So this is opposed to like a trans regulatory element, which might be something like a protein that binds to the DNA. This is actually on that same DNA itself. Kind of similarly, we have hetero and homo. So hetero means different or other. So something that's heterogeneous is going to be made up of multiple things and or not consistently distributed. So like it could be that you have different lots of things in there, or it could just be like one thing, but it's not really well mixed. Or we could talk about a heteromer, which is basically a complex with different components. And we have the opposite of that. We have homo, which means same. So homogeneous is going to be basically made up of a single thing and or something that's like consistently distributed. So it can be, you can have a homogeneous mixture, it just has to be like stirred up nicely. And a homomer would be a complex with multiple copies of a single component. So you might have like a homodimer or a homotetramer. So that would be like 2 or 4 of the same copies of like a protein that are working together. Okay, amphi, ampho, ambi, these mean both. So amphoteric is something that's able to act as both an acid and a base. Amphiphilic is having both hydrophilic and hydrophobic parts, and we'll get more into those words in a minute. But this amphi or this ampho, this is telling you it means both. So it can do both like different things. Okay, per means through. So like permeable is something that's permeable is going to allow things through, but will it allow all of them through? Introduce the term semi, which means partly. So something that's semi-permeable, like a semi-permeable membrane, is only going to allow some things through, but not others. And so a membrane might be permeable for little things, but not for big things and stuff like that. Pan means all. So if you talk about like pan genome analysis, this is basically they're telling you that their experiment was looking across the entire genome and not just at select parts. You might also see various pan transcriptome or whatever, like basically just all of whatever is after the word pan, the part pan. Holo means whole. So whole, holo. So you can probably figure that one out. So for example, a holoenzyme, this is going to be an enzyme with any of the necessary cofactors, so any of the helper molecules it needs. Holoenzyme is going to be the enzyme with those cofactors. This is in contrast to apo, which means without or lacking. So an apoenzyme is going to be an enzyme without those cofactors that it needs. You might also just see the word apo used by itself when you're talking about if you have, say, an enzyme without its substrate, you might call that apo. Whereas if you had the enzyme with its substrate, then you would just say enzyme. This is used a lot when you're talking, when like people are solving structures of, say, an enzyme or something else that has a ligand, that has a binding partner, and apo would be in the absence of that binding partner, and as opposed to having that binding partner, where we might refer to it as like a holo complex or things like that. Neo, this means new. So gluconeogenesis is the making of new glucose from non-sugar parts. So not breaking down glycogen or anything, but actually making new glucose from scratch, kind of. Well, not scratch, but it's from smaller parts. This is in contrast to paleo, which means old. So like paleogenomics, the study of ancient DNA, the whole Nobel Prize that was recently given. Okay, pro, before or in front of. So a prodrug, this is a word you might see. This is basically an inactive version of a pharmaceutical compound that's metabolized in the body to form an active compound. So sometimes the drugs themselves aren't very like bioavailable or whatever. Like if you were to try to give that drug itself, it might not be stable enough, or it might not be able to get into cells. Whereas if you give a prodrug, well, this might be not active by itself, but the body is going to metabolize it. So basically the liver makes some change to it, usually, that then makes it into the active compound once it's in the body. So you would have this version that's before that, or in front of the actual thing, like the real drug. Speaking of like real, U means true, normal, good. Eukaryotes, those are organisms whose cells have a true nucleus. So karyo is referring to nucleus, and then you have a true nucleus, so you have a U. This is as opposed to pseudo, which is fake or false. So you might see the term pseudogene. These are basically remnants of a gene that remain in the genome, but they aren't functional anymore. It turns out that a big part of our genome is actually non-functional stuff, or at least stuff that we're not sure what actually does. So that's a big part of it, and that Speaking of big, okay, that was a bad segue, but macro means big. So for example, macromolecules. These are large molecules, including nucleic acids, proteins, lipids. You might see the term macroscopic, which is something that you can see with the naked eye. This is as opposed to micro, which means small, or it means 1/1,000,000th if you're, we're going to our like SI prefixes. So like a microliter would be 1/1,000,000th of a liter, but you also see this outside of the term where it's not literally meaning 1/1,000,000th, but it's meaning small. So microscopic is something that you need a microscope to see. If you go even smaller, now we're talking nano, so this is super duper small, or 1/1,000,000th. So and then in the metric prefixes, or basically what people put in names to sound cool. So sometimes people just put like nano something or another, and they just mean that that thing is really small and it's cool. And so you might see nanometer, nm. This is basically a billionth of a meter. Nano discs, these are cool. These are tiny little discs that you like, tiny lipid discs that you can surround membrane proteins in to try to like visualize those proteins with techniques like cryo-EM. But basically, they're small and they're letting you see small things. Co means with or together. So, like I've talked before about co-precipitants. These are things like glycerol or glycoblue, which is a di attached version, so di-conjugated form of glycerol that you can add to help nucleic acids precipitate. So basically, it'll precipitate with them, helping draw those nucleic acids out of solution. It's really great if you don't have that much nucleic acid, but it's co-precipitating, so it's coming together. Kin means move, so like kinetics. This is the study of the rate at which things such as enzymatic reactions happen. And when we talk about a kinase, that's basically an enzyme that moves phosphate groups from ATP to other molecules. So kine or kin moves. Where are you moving? So peri means around. So periplasm is the space around the bacterial cytoplasm. So the cytoplasm is like the general interior of the cell. And then these bacteria have these like two membranes, well, at least some types of bacteria have these two membranes. And between those inner and outer membranes is going to be the periplasm. And it's basically around that cytoplasm. Meta means beyond. It's not just a Facebook company. Metastasis would be the spread of cancer beyond its original location. And so we'll talk about how stasis means kind of like place or staying in place. And so this would be spreading beyond. Telo means end. So telomeres are basically the end parts of chromosomes. And there's some cool biology going on there that I'm not going to get into. But I will get into the roots of some other terms. Okay, so a lot of these can also be prefixes or suffixes, so I wasn't sure where to list them, so I just kind of put them all here. And there's kind of just like a wide range of stuff because we have a lot of terms because we need to talk about a lot of different things. So if you see Jane or Genesis, basically this is talking about birth or creation or making. So glycogenesis would be the making of glycogen, a storage form of sugar. And we saw before gluconeogenesis, which is when we're making new glucose. Okay, hydro, this is means involving water or hydrogen. So hydrolysis would be the breakdown of water. If we're thinking of lipo or lipo, this is basically involving lipids. So lipids are your fats and oils and waxes. You see the term lipophilic, this would be lipid loving, basically a molecule that associates with lipids such as membrane bound proteins would be lipophilic, as opposed to hydrophilic, and we'll get more into those later. Okay, if you see glycol or glyc, this is basically involving sugars or it's sweet. So glycolysis is the breakdown of glucose, and glycoproteins are proteins with sugar chains attached. We call this sugar chains glycans. There's your glyc. Saccharine is another term that means involving sugar. So you might see like monosaccharides. These are basically sugar monomers. And remember, monomers before, those were like individual subunits or individual units. Halo, this is involving salt. So we were sweet and now we're salty. So halo means, this is referring to salt. A halophile is an organism that lives in high salt conditions. So these are some types of extremophiles, so things that live in really weird extreme conditions, and some of them are halophiles, which means that they're living in high salt. If you see oxy, either with a Y or with an I. This is involving oxygen. So like oxidative stress is a type of cell stress that occurs because there's a lot of reactive oxygen species. So you might see this like ROS, ROS. So basically highly energy oxygen molecules that like to attack things. And so oxidative stress, oxidative, so it's involving the oxygen. Some other elements, sulfa, thio, mercapto, these are all roots or prefixes or whatever that you might see when something contains sulfur. So for example, some of the commonly used reducing agents in the lab, things like dithiothriotol, DTT, and beta-mercaptoethanol, BME. So you see thio and mercapto, those are referring to sulfur, as would sulfa. Ferro, this is involving iron. So you might have heard of ferritin, which is this protein that like binds, stores, and transports iron. More and more like popular these days, or not popular, but I guess they're learning more about it, and you seem to hear about it a lot, is ferroptosis, which is a form of iron-mediated cell death. You also see some terms with cupra or cupra. I don't know, it's hard to say these things, but you can see it. This is basically telling you it involves copper, and so you'll see this in various chemical names. You might also see heme or hem. This is relating to blood, so hematocytes are going to be blood cells, and we'll see later that cyte is just referring to cells. Okay, one that we came across before, therm. This is involving heat. So remember, mesothermic was living at moderate temperatures. So we had our meso, which was moderate, and then our therm involving heat. Speaking of heat, another word we have for heat, calor. So like calorimetry is measuring changes in heat to study the energetics of molecular interactions and reactions. So that's calorimetry. So calor. Don't confuse that with color. So color and chrome are referring to color. So basically, if you had like a colorimetric readout, that'd be like an experiment. We have a color-based readout, such as like something binds to a visible dye or a dye changes colors. These are things that you can see with your eyes, and you don't need to actually use a fancy machine. So we can also have photo, which involves light. So photosynthesis is the making of food using energy from light. Okay, what else? Stere and stereo, these are, or like steric, things like this, these involve spatial orientation. So stereoisomers are chemicals that have the same atomic makeup in terms of like what's linked to what, but they differ in which way those groups actually stick out in space. And so here you see a couple of different things we've seen. So we have stereo and we have iso. Remember, iso was going to be like similar or same. Okay, morph, shape. So morphology is going to be the shape, something shape, or it can be the study of shape because ology is going to be study. Hist and histo, this is involving tissues. Histology is the study of tissues. You might see like immunohistology or whatever. That's basically staining tissues with antibodies and things like that to look for specific things. But the hist part is going to be meaning it's done in tissues. Cyte, this refers to a cell. So like a cytoskeleton is going to be this like skeleton inside of your cells of these various proteins that are helping give the cells its structure and things around. We also see cyte in describing a ton of different types of cells, like erythrocytes, which are red blood cells. Okay, phago, this means eat. So phagocytosis is going to be, basically it's this cool way in which cells kind of like pinch in their membrane to take things, take like a big bite out of what's outside. There's also macrophages, which are a type of immune cell that eat foreign stuff. There's also pino, which means drink. So pinocytosis is basically another way that cells can like pinch in their membrane to take things in, but here they just take in like a smaller portion. And pino, you can think of like wine. I don't know, it seems kind of like wine. So Pinot Noir, is that like a wine or something? Is that why it sounds? I don't drink. But anyway, cells drink, and that's pinocytosis. Four, this is to bear or carry. So an ionophore is a molecule that helps transport ions across membranes where ions are charged particles. An ionophore is going to help those things get across the membrane, and they need help doing that because those membranes are like all lipidy and stuff, and so often they need help. And so an ionophore can help transport them. Okay, last but not least, we have some suffixes. So philic or phile, we've seen this a lot of different times. This was loving or lover. So hydrophilic is water loving or water loved. You're probably going to see this term the most, hydrophilic. Basically, it's technically loving, but it's more like it's water loved as well, because it's water that's often the more picky one, whether or not it'll actually break up its water networks in order to surround something. But anyway, lipophilic, this would be lipid loving or lipid loved. And then we talked before, we also mentioned a halophile. So this is the organism that grew in high salt conditions. So it loves that salt. The opposite of this is phobic or phobe. So technically this means fearing or fearer, but I like to think of it more in terms of just like avoiding or avoided. So hydrophobic would be water avoided or water basically not it's not that the molecules fear the water, it's just the water doesn't really want to hang out with them, and so it excludes them. But anyway, that's just like a little personal thing I have. Pathy, this is referring to disease. So a tauopathy is a disease characterized by misfolded and aggregated tau protein. So you'll see pathy at a lot of different disease names, but that is indicating it's a disease. Lysis, this refers to breaking or breakdown. So lysis, we can use that term itself. So this is when we just lyse open cells, we break them, or we can stick it onto the end of other words. So catalysis is going to be the breaking down of biochemicals for energy and our building blocks. We have hemolysis, remember heme with blood. So this was going to be the breakdown of blood cells. ACE, ASE, sometimes AZE, this has been basically an ending that denotes that something's an enzyme. So a nuclease is going to be an enzyme, so a reaction speeder upper. That in this case, what it's doing is it's catalyzing, it's speeding up the breaking up of nucleic acids. So a nuclease is going to cut nucleic acids. If you see the term omics, you'll see this a lot these days. Sometimes it's kind of like a running joke that you can just stick omics on the end of anything. Basically, it just tells you that there's like an experimental method or field that deals with gathering and analyzing huge amounts of data. So it's like big data stuff. An example, a common one is proteomics, which is the study of all the proteins made and how this changes under different circumstances, like looking at different tissue types, et cetera, and generating a ton of data, typically with like mass spec, and then doing a bunch of data analysis to compare them and things. Genomics does something similar, but they're studying and comparing genomes rather than studying comparing proteomes, but they can do like between different species and different cell types and things. But then you have omics of all sorts of different things. So you have like phosphoproteomics, which is just focusing on phosphorylated proteins. You have epigenomics, which is looking at epigenetics of genomes. You have all sorts of complicated things where they're generating a lot of data and doing comparisons. If you see trophy, tropism, this is having to do with eating, nutrition, and or growth. So we saw before autotroph, so auto was self, troph is having to do with like this eating and nutrition. So an autotroph is making its own food. See gram or graph, it's telling you something like a chart or a graph. So a chromatogram is a graph that's showing light absorbance corresponding to sample coming off a chromatography column. So when you're doing like a protein purification, as it's coming off, the protein's coming off the column, it's going through like a UV detector, and then you're seeing on the screen the UV trace as the things come off the column, and that would be your chromatogram. You see metry or meter, that's basically measurements. So mass spectrometry is a technique that measures the mass charge ratios of molecules. If you see OG, this is the study of, so biology, the study of life. Stasis is standing or in place, staying. So homeostasis is basically a state of equilibrium or balance where things are staying the same. So remember homo, same. Stasis, place, standing, staying, or whatever. So in homeostasis, things are kind of constant. Not necessarily that nothing's happening, but there's no like net change. And so things have kind of reached their happy place. speaking of happy place, I hope this post helped you reach your happy place. And yeah, so I'll post a link to where you can view and download and stuff this list, and I will keep adding to it as I inevitably think of more things. And this is not at all a comprehensive list, it's just some of the main ones that you might see.